Hello, my name's Stuart Cobb from the University of Edinburgh, and today I'm going to talk about genetic therapy approaches in Rett syndrome and give a general update on the field. So this first slide shows the sort of key milestones in Rett syndrome translational research from the initial description of Rett syndrome back in the 1960s to the discovery of MECP2 early in the 1990s to then the link between MECP2 and Rett syndrome in the late 1990s. And then since then, there has been the demonstration of reversibility of phenotypes in animals modeling Rett syndrome. And then in the last 10 years, there has been really a sort of explosion of research focusing on approaches to try and target the underlying cause of Rett syndrome. So in 2013, the first proof of concept papers for Rett syndrome were published. And 10 years later, you can see from the list of companies on the right-hand side here that there are multiple biopharmaceutical companies now very active in the Rett syndrome space in terms of developing disease-modifying therapies. So I'm going to start off talking about, what do we know now as a sort of prelude. So of course, we know what Rett syndrome is. We know what causes Rett syndrome. It's due to mutations in this protein within the nucleus of the cell called MECP2. And we now know what the main function of MECP2 is. So we know that for disease-modifying therapies, the challenge is, can you put back functional MECP2 protein or correct the mutations that actually cause the loss of protein function? And this is what I'm really going to focus on today. So the next question is, what have we learned from animal studies to date? And the answer is a lot. So on the left-hand side of this plot, or this slide, this is showing that Rett syndrome is likely to be a treatable disorder. So we now know from studies going back 15 years now that if you switch off the MECP2 gene in mice, the mice develop a range of phenotypes which are modeling those phenotypes seen in patients with Rett syndrome. But if one then switches the gene back on, restores MECP2, not only do you halt the disease progression, but you actually improve multiple phenotypes of the disease, multiple features of the disease. And basically this list here is a list of features that have been looked at. And the bottom line is that everything that's been looked at shows some potential for improvement, whether it's motor function or things like autonomic function in the form of breathing or apneas, or whether it's things like EEG activity or seizures. So the next thing that animal studies have really sort of confirmed is that the brain is the main target system for looking for therapies for Rett syndrome. So although the MECP2 protein is actually expressed throughout the body, it's in pretty much all cells throughout the body, it's really a lack of MECP2 within cells in the nervous system that is driving Rett syndrome. And therefore, any therapeutic is really aimed at delivering MECP2 or correcting MECP2 in the nervous system. And then finally, animal studies have also shown the importance of MECP2 levels. And this is really critical when thinking about therapeutics, because too little MECP2 function drives disease in the form of Rett syndrome. But we also know that too much MECP2 can also lead to severe neurological disease. And this has been shown in patients with the duplication syndrome, but it's also been studied systematically in experimental animals as well. So this again is showing that the correct levels of MECP2 are important. So translational efforts in terms of developing therapies for for Rett syndrome, there has been a sort of shift from a lot of early work looking at small molecules, or what's called more conventional pharmacology, to therapies that are targeting really the root cause of the disease, which is mutations in the MECP2 gene. So the left-hand side really shows all the different pharmacological approaches and pharmacological agents that have been tested in Rett syndrome. And the bottom line is that the vast majority of these studies have been disappointing in terms of their outcome. The one exception here is trofinetide, which is likely to be an approved drug soon for Rett syndrome. That's an IGF-1 tripeptide. But really what I'm going to focus on is the right-hand side here. So therapy is targeting the root cause of Rett syndrome. So these are therapies that are either replacing or correcting the gene at the level of the DNA, or correcting mutations at the level of the RNA, or delivering protein, MECP2 protein. So I'm not going to talk about the last one. So conventional gene therapy, which is sometimes known as gene transfer or gene replacement therapy, is really a fairly simple concept. So where you have mutations that cause loss of function or loss of the presence of a gene, then you can deliver a therapeutic gene, what's known as a transgene, to replace the missing gene. And this is the case in Rett syndrome, where you are basically restoring a working copy of the MECP2 gene. And of course, as I've mentioned, it's important to be able to replace that to cells in the nervous system. So a number of us have been now working on this for many years. As I mentioned, the initial proof of concept studies were published exactly 10 years ago now. And this basically showed that if you took the MECP2 gene sequence and you packaged that into a viral vector, which is the basically the entity that allows you to deliver the gene to the body, and you use that and you treat animals that are modeling Rett syndrome, in this case the most common mutation causing Rett syndrome, what you get is you get a reduction in the severity of the neurological features when you deliver this gene therapy product. And what we find from lots of studies and multiple groups is that basically you can see a therapeutic effect, but the reason it's taken a long time is that we've wanted to develop better gene therapy molecules with both improved effectiveness or efficacy, but also better safety feature. Because what's been very clear is that there have been a number of reports showing that the safety window for Rett syndrome is very important, and one needs to put back appropriate levels of MECP2. So this is just to give a couple of examples. For instance, this was a paper that was published a couple of years ago now, showing that if you sort of overshoot and give too much MECP2, you can get a range of adverse effects. And similarly, we had shown also that you need to manipulate the gene therapy cassette in order to get something that's safe and effective. So I'm going to talk about more of this from a neurogene perspective shortly, but what I'd like to next go on to is talk about what's new or recent in terms of genetic therapies for Rett syndrome. So the first thing to say is that there's a lot of activity. So this slide is taken from the Rett Syndrome Research Trust, who have really spearheaded genetic therapies in Rett syndrome and funded a lot of the foundational work in this area. And what it's showing is that with conventional gene replacement, gene therapy, there are multiple companies and academic groups working on this. The two most advanced are Taysha and Neurogene, which are going into clinical trials this year. There's also groups looking at MECP2 reactivation, so that is switching back on MECP2 on what's known as the inactive X chromosome. Come on to this shortly. And then there are a bunch of labs and companies that are working on editing Rett-causing mutations at the level of the RNA, and further companies working at mutations at the level of the DNA. So I'll just give some examples of these shortly. So this section's who's doing what in the field. So this is a bit tricky because the majority of genetic therapy efforts are really being conducted by companies operating in stealth mode. So there's not actually all that much information out there in the public domain. So the four companies that I've sort of highlighted here with the red circles are the companies that have actually shown publicly some data for the therapeutics that they're developing. But the majority of the companies are operating in stealth mode and have not shown it publicly. So the first example I'll give is the work of Gail Mandel's lab, and they've really been pushing the boundaries in terms of what's known as RNA editing, and this is showing some promise. So basically what they're doing is that they're attempting to correct Rett-causing mutations at the level of the RNA, and this is very attractive because if you can correct the mutation, in theory you can correct specifically the cells that are expressing the faulty copy of MECP2. So this is a very attractive feature of editing approaches. And the other attractive feature is you can't really overshoot because as you correct the mutation, the MECP2 would then be expressed at the normal levels in the correct cell types. So how's she done this? So this is a paper that was published very recently in which Gail's lab has shown when they deliver their therapeutic molecules here, they can achieve editing of a specific Rett-causing mutation. So they were looking at a very specific mutation, and this is the model, this G311A model of Rett syndrome. And the data showing here is that these mice develop apneas, which is a characteristic phenotype, a cardinal phenotype seen in many Rett patients. And this is seen in the mice as well. So these sort of gray, these pauses here are apnea events in the untreated disease animals. And you can see that following virus treatment, this respiratory pattern has been normalized and the number of apneas is greatly reduced. So this is the untreated animals here. You can see there's high number of apneas in many of the animals, and in the treated animals, this has gone back down to very, very low levels, similar to normal healthy mice. So this is very encouraging. Other data that's been presented in the public domain is this other approach, this what's known as X chromosome reactivation. So there's a company, Alcyone Therapeutics, who are pursuing this approach, and their drug, which is ACTX-101, is essentially an adeno-associated virus. So it's a gene therapy virus, but instead of delivering a therapeutic gene, they're delivering what's known as a microRNA sponge. And the idea of that is to really switch back on the healthy copy of MECP2 that is in the cells that basically normally lack MECP2 function in girls with Rett syndrome. So they have presented mouse data at scientific meetings. It's not been published in papers yet, and they've also shown safety data in large animal species. So finally, there's also a lot of progress being made on RNA editing, and in particular, there has been data presented on what's known as read-through of premature stop codons. So as you're probably aware, many cases of Rett syndrome are caused by what are known as premature stop codons. So basically any of these that end in an X, any mutation that ends in an X has a premature stop codon. And basically this therapy works by allowing you to read through this premature stop codon. So instead of getting so if you get a premature stop codon mutation, what you end up with is a truncated, in other words, inactive or reduced activity form of MECP2, but using these basically these read-through molecules, which is what's called a tRNA molecule, they can facilitate reading through to produce normal functional MECP2. So there was a lot of work done on this a number of years ago with small molecules, but the results of those ended up being somewhat disappointing. And what the companies working in this area are claiming is that they get much more effective read-through of these premature stop codons. And there's been some data presented in this R255X model. So I would lastly just like to say that there's also very intensive efforts to develop better ways to actually deliver these genetic payloads in terms of these therapies. So one of the challenges is actually getting these genetic therapeutic agents into the nervous system. And most of the approaches to date are delivering directly to the nervous system. So for instance, the Neurogene studies delivering to the fluid-filled ventricles in the brain, the Tatia studies delivering to what's known as the lumbar spinal cord, and there are other companies that are working using this, what's called ICM route of delivery. But there is a definite requirement to get more effective delivery systems to the brain. And again, there are a lot of companies working on developing new viral vector capsids that can make it more effective to deliver to the nervous system. And ultimately, this will benefit Rett syndrome because the brain is the key target. So to conclude, loss of function of MECP2 is the root cause of Rett syndrome. And there are multiple animal studies from across multiple labs that predict that Rett syndrome's a treatable disorder if you can actually restore MECP2. And the last few years have seen a great deal of activity in developing what are likely to be disease-modifying therapies. And the three that sort of stand out are gene therapy, X chromosome reactivation, and RNA-level editing. And then my last point was really, with all of these, delivery to the brain remains a challenge. And, for a Rett syndrome therapeutic to be truly effective, we really need to maximize delivery to the nervous system. But with that, I'll stop and thank you.